Hello everyone. Today we are going to solve this paper of NEST 2021, at least some of the questions, especially the biology section. So before solving this paper, let us first see what were the main chapters that were covered in this paper, or that were questions were asked from those chapters. So we predicted earlier NEST 2021 that which chapters are important. And luckily questions from those chapters were asked also. So these chapters are, first of all, molecular biology is the most basic chapter that has been asked. Either interrelated question will be asked or directly question will be asked from these chapters. Molecular biology, which includes your DNA part, all those things, DNA, RNA. Second thing is biotechnology. So both of these chapters are related because both of them plays with DNA and RNA. Now the third chapter that is very important is cell biology, and I'm also including microbiology in it, which includes bacteria and all those things. These third point, cell biology and microbiology, it covered at least 4 to 5 questions on this paper, so it is very important chapter. Now the fourth unit that was being covered was ecology, and I'm also including evolution in it. Again, evolution se question jo aate hain, they are interrelated, not direct ones. Also, the questions from classical genetics was asked. Classical genetics means the one which includes your Mendelian laws. Or one last important chapter that was being asked was plant physiology. Plant physiology is also very essential with respect to this exam. Also, some parts of metabolites, metabolism part, or we can say enzyme activity and all those things were asked. Okay, so it also includes biochemistry. So if you are thorough with these 7 units, I'm sure that you will be doing very well in this exam. And thorough means not just about reading the chapters. It is, by thorough I mean that you have understood it conceptually and you are able to interrelate all these chapters. Then only you will be able to solve all the questions of this paper. So let us see some of the questions. Like this first question, I will not read the whole question. I will just tell you what the question is asked. This question में कह रहे हैं ये लोग कि a group of scientists that you all are going to be researchers, they wanted to study the ancestry or common ancestry between 2 species. Let us say we have species P and species Q, and we want to study that whether P and Q species have common ancestry or not. मतलब कि उनके parents किसी एक टाइम पे सेम थे या फिर डिफरेंट थे, ये स्टडी करनी थी. एंड इन ऑर्डर टू डू दैट, what they did was that they sequenced the genetic content, that is DNA and RNA, and also they sequenced amino acid sequences of some of the proteins. ठीक है, amino acid sequences of some proteins, और इन दोनों के बेसिस पे वो इन स्पीसीज जो कि P and Q है उनके बीच कॉमन एंसेस्ट्री फाइंड करने की कोशिश कर रहे हैं. ठीक है, so this question is actually interrelated. So we have to cover 2 chapters in order to solve these questions. First one is molecular biology and second one is evolution. जो हमारा ancestry है वो define कौन करता है? It is defined by the genetic elements, and genetic elements you all know that it is DNA and RNA. So if DNA or RNA sequences are same or similar in both the species, that is P and Q, then we can say that they follows common ancestry. Okay, this is one point. And protein sequences agar same hai, then it can have same ancestry, but protein sequences, the amino acid sequences का same होने का मतलब है कि they will have similar function. ठीक है, so if protein sequences are same, then we can say that these 2 proteins will have common functionality. So protein sequences signifies functionality, while this thing, genetic content, is signifies common ancestry. ठीक है, और evolution में तुम लोगों ने पढ़ा होगा कि there is something known as homology and analogy. Homology means common ancestor or same structure but different function. Same structure of particular organ having different function. Different functions. And analogy means ancestry है वो differ करती है. So we can say that not common. Ancestors are not common. are not common. And second thing is that although the ancestry is not common, but still that organ which we are studying, it is showing same function. Or homology is also known as divergent or divergence. Okay? Because homology means what? Same origin but showing different function, which means that it is undergoing divergent evolution. It is evolving, it is actually diverging. Okay? So it is an example of divergent evolution. Now analogy means same function but different origin. It means that it is converging toward one point. So it is an example of convergent evolution. Now, knowing these two facts or these two information, we have to tell that which among these options is correct. Okay. Now, the first statement is saying that similarity in exons but not necessarily in introns is reflective of divergent evolutions. Correct? Because exons are the coding sequences. So we are more concerned about the similarity in the coding sequences, which are coding for a polypeptide. Introns are also our concerns, but even if they are not same, then also we can say that they are or they belong to same ancestor on the basis of similarity in exons. Okay? So this statement is correct. Now second statement is saying that similarity in the protein sequence but not in the corresponding RNA or DNA sequence can be an example of divergent evolution. So divergent evolution means what? Common ancestry. And I have already told you that common ancestry is tracked by DNA and RNA sequences, not the protein or amino acid sequences. So which means that similarity in protein sequences is actually responsible for functionality, but not common ancestry. Okay, so that's why this statement is wrong. Now the third statement is saying that similarity in introns but not exons is reflective of convergent evolution. So first and third statement are what? They are working against, they are antagonistic to each other. Okay? So introns are actually non-coding sequences of DNA. So हमें non-coding sequences से कोई concern नहीं है. We are concerned about the coding sequences. Okay? So similarity in introns but not the exons is reflective of convergent evolution. This statement is obviously not correct because we are concerned with similarity in exons. If introns are also similar, then it is good. But even if they are not similar, then also it is fine to go with exons. Okay. Now, fourth statement. Though the overall protein sequences are not similar, the conservation of active site residues cannot be due to convergent evolution. So they are saying that अगर एक protein है, it has different amino acid sequence, but active site पे उनके sequences same है. Active site पे उनके sequences same है, तो वो convergent evolution show कर रही है. इस स्टेटमेंट बोला है कि ये convergent evolution नहीं show कर रही है. ये इस चीज का एग्जांपल है. But it is not correct because the main functionality of protein is determined by the active site. So even if the whole protein, the whole amino acid sequence is different and only the sequences of the active site are same, then also both the proteins will be performing same function or similar function. Okay? So this statement is also incorrect क्योंकि यहाँ पे cannot be लिखा हुआ है. It should be can be. अगर cannot की जगह can होता, then this statement would have been correct. Okay, so this statement is incorrect. तो अभी देख रहे हो तुम लोग कैसे ये फर्स्ट क्वेश्चन है जो कि हमारा दो चैप्टर्स की अंडरस्टैंडिंग को चेक कर रहा है. Now let us see the second question. Okay, again, I will not read the question. I will just say what the question is saying. So the question is saying that there is some person known as Mr. P. Mr. P is not able to father a child because of one defect in its mitochondria. तो क्या है इसमें डिफेक्ट? डिफेक्ट यह है कि its mitochondria has undergone deletion of some DNA sequences. ठीक है? Has undergone deletion, which is a kind of mutation. Deletion in DNA sequence of mitochondria. So mitochondria टर्म को तुम लोग याद रखना कि ये जो म्यूटेशन हुई है ये माइटोकॉन्ड्रिया में हुई है और कहीं नहीं हुई है, नॉट इन द सोमैटिक सेल, नॉट इन द एक्स क्रोमोसोम्स. ठीक है? तो अब उसकी माइटोकॉन्ड्रिया में कुछ डिलीशन हो गया है. नाउ ड्यू टू दिस डिलीशन द mitochondria of the sperm, they are not able to synthesize ATP. Able to synthesize ATP. They are not able to synthesize ATP. और ऐसा क्यों हो रहा है? क्योंकि ये माइटोकॉन्ड्रिया में जो जीन अफेक्टेड है, जो जीन में सीक्वेंसेस डिलीट हुए हैं, इट वाज कोडिंग सम प्रोटीन X which was responsible for the production of ATP. ठीक है? अब वो X डिफेक्टेड फॉर्म हो रही है और डिफेक्टेड X फॉर्म होने की वजह से देयर इज लेस प्रोडक्शन ऑफ ATP वी कैन से. एंड वी नो दैट द स्पर्म इट हैज abundant amount of what mitochondria in its neck piece, है ना? ऐसे स्पर्म होता है, यहाँ पे उसका मिडल पीस होता है, एंड दिस मिडल पीस has abundance of mitochondria. And why it has abundance of mitochondria? Because sperm has to move in order to approach ova. ठीक है? For movement. ठीक है? अब वो मूव नहीं कर पा रहा है. Why? Because it is not able to produce ATP. And ATP, we know that it is essential for what? For energy production. और अगर एनर्जी ही नहीं मिलेगी तो sperm मूव नहीं कर पाएगा. ठीक है? तो अब वो मूव नहीं कर पा रहा है, इसकी वजह से he is not able to fertilize an ova. ठीक है? And hence, if it is not able to fertilize an ova, then zygote will not form, and if zygote will not form, then embryo will not form, and subsequently the child will not form. No child. Very sad. ठीक है? तो अब इस डिफेक्ट को या फिर इस कंडीशन को सॉल्व कैसे किया जाए? इस कंडीशन को कैसे सॉल्व किया जाए? देखते हैं कैसे सॉल्व कर सकते हैं हम इस कंडीशन को. इस कंडीशन को सॉल्व करने के लिए हमारे पास असिस्टेड रिप्रोडक्टिव टेक्निक एम्प्लॉय करने का तरीका है. ठीक है? Assisted reproductive technique, or we can say ARTs. They are widely used in modern day sciences. तो क्या किया जा सकता है? अब प्रॉब्लम क्या है? प्रॉब्लम ये नहीं है कि स्पर्म या उसके जेनेटिक्स में प्रॉब्लम ये है कि उसके माइटोकॉन्ड्रिया के जेनेटिक्स में प्रॉब्लम है ना? और माइटोकॉन्ड्रिया के जेनेटिक्स में प्रॉब्लम है जिसकी वजह से माइटोकॉन्ड्रिया इज नॉट एबल, स्पर्म इज नॉट एबल टू मूव, और मूवमेंट किससे चाहिए थी? क्योंकि उसको ओवा को अप्रोच करना है, which will be present in which segment of female reproductive system? It will be present in the ampullary isthmatic junction, है ना? वहीं पे फर्टिलाइजेशन होगी. तो हम अगर फीमेल के ओवा को रिप्रोडक्टिव ट्रैक्ट से निकाल ले और मिस्टर पी के ओवा स्पर्म को भी निकाल ले, and we fertilize it in the test tube or in vitro conditions, then surely it will form a zygote, है ना? So if it is done in vitro condition that we usually call what? Test tube baby, then this problem will be resolved, है ना? क्योंकि प्रॉब्लम जेनेटिक्स में नहीं है, प्रॉब्लम है स्पर्म के मूवमेंट में. स्पर्म मूव नहीं कर पा रहा है तो हम उसको मूव करा के भेज देते हैं. So it will be resolved by doing in vitro fertilization. And after in vitro fertilization what we should do? अब जाइगोट तो बन गया है, लेकिन जाइगोट का डेवलपमेंट जो है, एम्ब्रॉय का फॉर्मेशन, फीटस का डेवलपमेंट, ब्लास्टोसिस्ट का बनना, ऑर्गेनोजेनेसिस होना, ये सब प्रोसेसेस कहाँ होती है? इट इज पॉसिबल ओनली इफ दिस जाइगोट इज ट्रांसफर्ड टू फीमेल वॉर्म, है ना? सो वी हैव टू fertilize it in the in vitro condition, and then we have to subsequently transfer it to the female's fallopian tube if it is in early stage of development, or else if it is in later stage of development, then we have to directly transfer it to the uterus where it will undergo implantation and subsequently it starts its development, है ना? और इस प्रोसेस को हम बोलते हैं zygote intrafallopian transfer. क्या किया जाता है? जिफ्ट में जो फर्टिलाइज्ड zygote है उसको लेकर फीमेल के fallopian tube में इंजेक्ट कर दिया जाता है. यही इसका आंसर भी है क्वेश्चन का. दैट इज B option. The B option says that an intracytoplasmic sperm injection procedure followed by a zygote intrafallopian transfer with help of Mr. P will help Mr. P father a baby. Correct है ना, क्योंकि वो मूव नहीं कर पा रहा था इसलिए हमने Mr. P के एक sperm को लिया and we injected it in the ova in vitro condition, which is known as intracytoplasmic sperm injection, which is also called ICSI, and subsequently ZIP, zygote intrafallopian transfer. ठीक है? So this question was quite simple. If you have read which chapter, reproductive health. Now let us move on to third question. So third question is linked with which chapter? Genetics, or more appropriately, classical genetics. So if you have read about Mendel and Mendelian laws, then you will be surely able to do this question. Now let us see what the question says. Question is saying that in some variety of dog, the skin coloration, it is governed by two genes. First gene and second gene. And they are saying that the first gene, it has two alleles, capital P and small p, and the second gene has also two alleles. Allele तो दो ही होंगे, capital Q and small q. But what is the function of this gene and this gene, first and second? So first gene is actually leading to the color production. Okay? So its function is color production, and gene 2 is leading to what? Color deposition in the skin. So deposition. And in the question it is written that this capital P leads to black color, leads to production of black pigment. Small p leads to production of brown pigment, and capital P is dominant while small p is recessive. And here also capital Q is dominant and small q is recessive. और कैपिटल Q से होता क्या है? अगर कैपिटल Q जीन होती है, एलील होती है, ये जो भी कलर है ब्लैक एंड ब्राउन, इट स्टार्ट्स टू डिपोजिट ऑन द स्किन. ठीक है? सो कैपिटल Q लीड्स टू डिपोजिशन ऑफ कलर, while small q leads to what? No deposition of color. No deposition. अब अगर कुछ भी डिपोजिशन नहीं होती है कलर की, तो चाहे तुम्हारे पास कैपिटल P है, चाहे तुम्हारे पास स्मॉल p है, कोई डिपोजिशन नहीं होगा. ब्लैक कलर भी नहीं देखने को मिलेगा, ब्राउन भी नहीं देखने को मिलेगा. तो क्या मिलेगा? In the question it is written that in such condition we are seeing albino color. ठीक है? तो हमारे पास तीन possibilities है. Either black color will be formed, black colored dog will be inherited, or brown or albino. ठीक है? अब देखते हैं कैसे इस क्वेश्चन को सॉल्व करना है. क्वेश्चन में कह रहे हैं कि if two varieties or two organisms, one male and one female, having heterozygous condition for both the genes are mated, then what is the probability of these colors to be seen in the progenies? ठीक है, तो देखते हैं. तो यहाँ पे भी हमारे पास कैपिटल P स्मॉल p, कैपिटल Q स्मॉल q है. फीमेल से भी हमारे पास कैपिटल P स्मॉल p, कैपिटल Q स्मॉल q. Now you all know that we have to apply Mendelian genetics here. कैसे अप्लाई करेंगे? So we will first see what are the possibilities of different kinds of gametes from this genotype. So it can be capital P, capital Q, capital P small q, small p capital Q, and small p small q. तो देखते हैं क्या होगा. ये चार possibilities हैं in male and in female also. Now let us draw a Punnett square and see how it goes. So capital P, capital Q, capital P small q, small p capital Q, small p small, capital P capital Q, capital P small q, small p capital Q, and small p small q. Okay, let's say it to be a male, let's say it to be a. इनसे progenies क्या क्या आएंगी? We will be able to see capital P, capital P, capital Q, capital Q, small q, and like that you have to draw all the possibilities, and at the end we have this thing. Okay, so in this question it has been written that small q is recessive, and if there will be this condition, which means that the genotype is small q, then surely it will give albino color, है ना? तो albino color आएगा. देखते हैं कब. अब small q वाले कितने cases हैं? We have this case. Let us use some other color. This one small q, this one small q, and where is the other? This one and this one. So we will have 4 albinos And which option among these four have 4 albedos? This one. Enough. So we know that this option is going to be correct. But let's see the other parts also. So if there will be only one P and only one Q, not only, at least, if there will be at least one P and one Q, then it will going to give which one color of dogs? Will give black colored dogs. इसमें black आएगा. This one will also give black. This one also. This one also. So 4 blacks from here. 5, 6, 7, 8, and so 9. 9 blacks will be there, and remaining will be brown. Okay, so 9 plus 4, how much? 13. So 16 minus 13 is equal to 3. So 3 will be brown. So this was the question. And if you have read what? Recessive epistasis, then this question will be nothing for you. Okay? The recessive epistasis का example है color, different colors of dogs in some particular species. Now let us see some other questions. Like this question. Again, I will not read the question. I will just explain you what the question is. Question क्या कह रहा है? Question कह रहा है कि there is one fungus which infects leaves of particular species of plant. And how does it infects the seed, the leaves? It secretes a chemical, and this chemical leads to prolonged opening of stomata. और stomata open होता है, उसकी वजह से stomatal pore भी open होती है, and this fungus infects the leaves via these pores. ठीक है? So they enters via pore. ठीक है? ये chronology है. अब वो लोग पूछ रहे हैं कि how is it possible? What will be this chemical doing so that this stomata is opening for a prolonged time? तो हम लोगों ने एक concept पढ़ा था in the chapter transportation in plant about opening and closing of stomata, and it was regulated by which ion? It was regulated by potassium or K plus ions. So I have told you that whenever K plus ions starts to enter the guard cells, what happens? The osmolarity of cells of the guard cells increases, and due to this increase in osmolarity, what happens? Rapid amount of influx of water happens, which leads to increase in turgidity of guard cells and hence opening of stomata for a longer period of time. ठीक है? So अगर potassium ions prolonged time तक import होते रहे, then it will cause prolonged opening of stomata. कौन सा option सही हो रहा है इस concept को apply करते हुए? This fourth option. Clear? Now let us see the fifth question. So fifth question is nothing but if you know about the layers or number of layers in cell organelles or different cell organelles. So this question is saying what? It's saying that there is one protein named X, one protein named Y, and one protein named Z. So someone or some researcher like you tried to study the movement of these protein inside these cell organelles from the cytoplasm inside the cell organelles. Okay? And with time they found that X was localized in mitochondria, Y was localized in endoplasmic reticulum or ER, let's say, and Z was localized in nucleoplasm, which means inside the nucleus. Nucleoplasm. अब वो nucleoplasm में है. Now they are saying that, or they are asking that, how many cell organelles they need to cross in order to be present in these organelles. तो अगर तुम लोगों को पता है कि ये organelles कितनी membranes comprise करती हैं, तो तुम लोग easily solve कर दोगे. जैसे कि if I say or if I ask you that how many layers are present on the outside of mitochondria, or how many membranes are there in the mitochondria, you would say 2 layers, or it is bilayer. And in endoplasmic reticulum we have 1 layer. In nucleus we have 2 layers. So now tell me how many layers they have to X, Y, and Z have to cross before entering into these cell organelles. So mitochondria में जाने के लिए it will have to cross 2 membranes or 2 layers. In ER it needs 1, and in nucleoplasm it needs 2. So knowing this information, knowing this fact, we come to this conclusion that while X and Z have to cross 2 bilayers, Y has to cross only 1 bilayer. Okay. So this was your fifth question. So these are some of the questions that I wanted to discuss with you all. Thank you.